Hi, this is Mr. Denver Teo, and I'll be using this schematic diagram as well as this video to illustrate the linkages between ETC, proton pump, and how all of this eventually translates into ATP synthesis. Before I move into detail about oxidative phosphorylation, let me first introduce to you this schematic diagram and also recap with you the important components involved in oxidative phosphorylation. Over here, you see this membrane is known as the inner mitochondrial membrane. The area below is known as the mitochondrial matrix. The area above is known as the intermembrane space. As you can see, there are four complexes: complex 1, complex 2, complex 3, and complex 4. Complex 1 stands for NADH dehydrogenase. Complex 2 represents succinate dehydrogenase. Complex 3 represents cytochrome BC1 complex, and complex 4 represents cytochrome C oxidase. Ubiquinone over here is a shuttler protein which shuttles electrons from complex 1 to complex 3. You can see the brown beads over here. The brown beads actually represent hydrogen ions. And I'm sure you're familiar with ATP synthase. This is the enzyme which is responsible for the synthesis of ATP from ADP and phosphate. You can see this pair of red beads over here. They represent a pair of oxygen molecule. And over here, you can see FADH2. This represents reduced flavin adenine dinucleotide, and it's carrying a pair of electrons. So the black beads actually represent electrons. NADH is known as reduced nicotinamide adenine dinucleotide, and it's also carrying a pair of electrons. So first, let's begin with NADH. NADH would carry electrons to complex 1 first. Upon reaching complex 1, the electrons would then be transferred from NADH to complex 1. Upon this transfer, NADH would then be converted into NAD+. So this is the regeneration of NAD+. The pair of electrons would then be transferred from complex 1 to ubiquinone. Upon this transfer, it causes a conformational change in complex 1, which would allow 4 hydrogen plus ions to move from the mitochondrial matrix into the intermembrane space, as shown. 1, 2, 3, and 4. Okay? After which, ubiquinone would then shuttle this pair of electrons from complex 1 to complex 3. Upon reaching complex 3, the pair of electrons would then begin to move from complex 3 to complex 4. During this movement, it will also cause a conformational change in complex 3. So it will also prompt the hydrogen plus ions to move. Okay, and with this, additional 4 hydrogen ions will move. 1, 2, 3, and 4. So in total, there are already 8 hydrogen plus ions that are being transferred from the mitochondrial matrix into the intermembrane space. Okay, but while it's transitioning from complex 3 to complex 4, it will travel one at a time. So the first electron would be transferred from complex 3 to complex 4, and then it will allow the passage of 1 hydrogen ion into the intermembrane space. The second electron would then move to complex 4, and you will also allow the passage of another hydrogen ion into the intermembrane space. So in total, with a pair of electrons from NADH, it can actually allow the passage of 4 plus 4 plus 2, which in total is 10 hydrogen ions into the intermembrane space. And because of this introduction of 10 more hydrogen ions in this intermembrane space, you cause a slight rise in charge, because after all, hydrogen ions are positively charged in nature. You also cause a reduction in pH, because H+ will then contribute to a more acidic environment. As such, with this slight rise in charge and reduction in pH, this is actually a generation of the electrochemical gradient. So since there are additional 10 hydrogen ions in the intermembrane space, this would provide the proton motive force, which is important for chemiosmosis. So chemiosmosis is the process of converting this proton motive force into ATP synthesis via the action of ATP synthase. And how does ATP synthase work? Okay, so since there are 10 hydrogen ions, in total, all of them would go down back into the mitochondrial matrix via the ATP synthase pathway. 3 hydrogen ions equals to the synthesis of 1 ATP molecule. So as you can see, this tree will move down like this. And when this tree moves down, it will prompt P and ADP to combine to form ATP, 1 molecule ATP. Okay? Likewise, so on and so forth. So another 3 will go down and they'll combine. Right? Combine like this. And the last 3 or 4 will also go down like this. And it will lead to the combination of the third ATP molecule. So as you can see, one NADH with 1 pair of electrons will lead to the synthesis of 3 ATP molecules. Okay. Now let's take a look at FADH2. FADH2 doesn't go to complex 1. It is different from NADH because it will go to complex 2 instead. So you upon transferring the electrons from FADH2 to complex 2, as shown, FADH2 would then be converted to FAD, so it will result in the regeneration of FAD. With this pair of electrons would then be transferred from complex 2 to complex 3 over here. With this transferring, again, the same process occurs. Right, 4 hydrogen plus ions will be transferred. So I'll show 1, 2, 3, and 4. Okay, and the same process will happen as well. Okay, note that the electrons from NADH, they are still here. Okay, but this is the electron from FADH2. The first one will transfer from complex 3 to complex 4. You cause it to open, and 1 would enter the intermembrane space. The second electron would then be transferred from complex 3 to complex 4, and another hydrogen ion would be transferred to the intermembrane space. So in total, there are actually 6 hydrogen ions being transferred from the mitochondrial matrix to the intermembrane space from FADH2. Right, but because now this time there are only 6, so you can do the math. With 6, and it takes 3. Okay, so now let's redo the ATP. Okay, so now they're all back to ADP phase with the phosphate groups. Now ATP synthase will open up. The proton motive force would drive ATP synthase to synthesize ATP. So 3 at a time. The first 3 has gone down and you synthesize 1 ATP molecule. The next 3 will also go down and you synthesize the second ATP molecule. But note how many ATP molecules are being synthesized? Only 2, right? So as you can compare, a pair of electrons from FADH2 can only lead to the synthesis of 2 ATP molecules, whereas the pair of electrons from NADH, they can actually lead to the synthesis of 3 ATP molecules. And why is that so? It's because NADH goes to complex 1 first, and complex 1 is actually a higher level than complex 2. And because of all this, because there's an increase in the amount of electron transfer from complex 1 to complex 4, it will lead to the passage or the movement of more hydrogen plus ions into the intermembrane space, which would eventually translate into more ATP being produced. And notice how the electrons are still here. Okay, so this is where your final electron acceptor, oxygen molecule, comes into play. Okay, how does it come into play? The oxygen molecule right? One oxygen, just one oxygen atom, will combine with 2 electrons and 2 hydrogen plus ions to form water, right? The second one as well, one oxygen atom with 2 electrons as well as 2 hydrogen plus ions to form the second water molecule. And that is how ATP is being synthesized from the protein pumps and the ETC during the process of oxidative phosphorylation with the aid of chemiosmosis. Thank you.